honored and happy to be here in front of you, and I would like to thank each and every one of you for making this place like another home. I remember the last time we had been here was for a conference, and it was just before the lockdown. And thank God that many of us have been able to survive, and I also feel that much has to be learned during this COVID period, and it has made us more aware of how much health is important and nutrition as well. Yeah, I would like to thank the Honorable Vice Chancellor, first of all, for suggesting this idea of the endowment lecture. Although she could not be here today, I know we had met last time. And I also thank the registrar and the head of the departments, and also Professor Jnanam and Saranya in particular, and also Dr. Uma for a very nice way to host, and I continue to keep coming. With the MOU, I think no questions asked, we will keep coming. Yeah, so the testimony of this success of the MOU remains to be seen in the following years, but hopefully this is taking off really well, because I have some of my colleagues here who is going to help me to take a workshop in jointly with TNAU, as the registrar also pointed out. So I will start off the talk by giving you some introduction on how and why plants have been taking so much attention for human health, which are called as secondary metabolites, that have been observed in plants and have been exploited by human beings for many years. And in fact, almost as long as the humans have been here, we have been exploiting this. So what are the sort of proof that humans have been using these medicinal value of the plants? And let's take the very beginning, 1st century BC. Or the 2nd century BC, there have been texts in the form of these sort of, leaves, palm leaves, by which we have texts that are hidden into beautiful 3 big books, which are called the Charaka Samhita, Sanskrita Samhita, and so on. And the Charaka Samhita, for instance, the name suggests that Charaka means someone who's traveling. So essentially, this is knowledge that have been picked up by many travelers all around the world. It's not a single person called Charaka as we might think. In fact, it has been taken over from more people, and it has also been mentioned that the origin of Charaka Samhita is not exact, and in fact, prior to Charaka Samhita, we had something called Agnivesha Samhita, and that has been documented as something that started in the 1st or the 2nd century BC. And at that time, the next following traditions had to be kept, and some more people came in, and multiple authors tried to revive and hold the text. And it is mentioned that only 1/3 of the text could be found, and others have been either lost or destroyed, or simply we could not keep track of them. And therefore some of the authors have put in their own thoughts and tried to build them as what we see now as 8 books going into 120 chapters. And what do they convey? They talk about how plants are important for human health and how it has to be incorporated in our diet, hygiene, in prevention, and also in the medicinal medical education. And they make sure that the patient is able to recover in his or her health. And moreover, I would like to draw in the fact that it's not only in India, which we are happy and proud of about these textbooks, but also in Greek, Hippocrates had documented textbooks on plant as medicine as early as the 4th century BC, Aristotle in the 4th century BC, and also documents that are clearly available. And there have been very early realization, as in Charaka Samhita. What they say is the body is composed of molecules, and these are said to be numberless and numerous, and this is nothing but referring to the atoms, for which a scientific description or even definition had not yet set in. But the fact that the body is made up of so many millions of small molecules has already been realized in the 1st or 2nd century BC. So that is so humbling. And also in the 8th century BC, as of now, also we can find some kind of rock inscriptions in Indonesia, and wherein they talk about this sort of a huge tree, and there are lots of goddesses protecting their trees. And why? Because this is called as a kalpataru, and how people should protect it for their own welfare and so on. So these are all the ways by which people have tried to pass on through generations. Almost as long as the humans have been there, plants have been exploited by humans for their own health and benefits. And in fact, I also picked up another text because I am talking this in Tamil Nadu, where I belong. Let me also read it out for you. Aktipattai Churanam Seidu Nallannai Vittaraitu Pilavai Mudalanavarku Pattu Potal Gunamadam. So this is a clear kind of commitment, and there's a guarantee by which people say that if you apply a particular paste of the bark of a tree, you can get a cure from ailments such as skin diseases, or even something which may be more as much as ulcers and so on. So with this thought and a brief introduction to the importance of trees and how they are intimately connected with human health, let me also introduce to you the topic which is on the plant metabolites. Plants have to produce a lot of chemicals. Not only humans do, every living being have to produce chemicals for them to live and to be able to survive. And especially plants have to produce a lot more chemicals for them to survive, because they cannot move away from danger. They cannot move away from predators who might come and chew them, and they can also run away from biotic or abiotic stresses. Huge harsh sunlight may be there, and problems in the soil may be there, so they just have to deal with them then and there and make sure that they also survive in the process, right? So plants have produced what are called the secondary metabolites. As the name suggests, these are not required for the primary life, but they are there for as a defense purposes, and they might be producing this to make sure that the pests won't attack them, the fungi won't grow on them, the microbes won't take over them, or even the pests will not chew them up too much, right? So, and you can, if you go into the literature and start looking for the metabolites, as a very beautiful introduction that was given by the director of the PBMB today, you can see that several small molecules are endemic to different plants, specific for that plant, or at least isolated and purified or even recognized in that plant for the first time. So, for instance, Arabidopsis thaliana, you see that is a plant metabolite called thallianol. And then in lotus, you have a plant secondary metabolite called lotostyrene. And in tomato, you have something called alpha-tomatine. And then you have more and more compounds like this in different plants. And the list is simply endless. And therefore, like somebody said, even to choose what plants to study, what sort of trees to look in depth, it is a very hard decision, because almost every plant has a very beautiful story of many different secondary metabolites that they are producing all the time, and as I said, it could be for defense purposes. But also those who are introduced to a bit of chemistry will know that these chemicals are not simple. They are not at all simple. You can see that there are some thick bonds and then some dashes here and there. This suggests that some part of the chemical bond is in front of us if it is thick, and if it is behind us, it is shown as dashed. So with respect to the plane of the whole molecule, you can see sometimes chemical groups come in front and sometimes at the back. And if each of the chemical groups around an atom is different, then it's supposed to be a chiral molecule. Chirality has to be appreciated. not only because of the optics, but also because simply how complex that particular chemical is. The secondary metabolite is so complex that you can imagine, even if we had appreciated, for instance, the thallianol in Arabidopsis thaliana is important for a particular ailment, for a cure. Imagine if pharmaceutical industries have to synthesize that particular molecule with the correct chemical groups in front and back. It's going to be extremely challenging because you cannot produce these molecules in that sort of specificity and precision with the right chirality at different groups, and plants do them very well, and plants do them naturally, and therefore they are very, very smart. So this is what interested our group to participate and adopt plants as our genomes or as our organisms for genome analysis. And we chose medicinal plants because we know that all these secondary metabolites are synthesized within the plant by means of enzymes, and the enzymes ensure that they provide the right sort of chemistry and environment so that the right molecule is produced and the wrong molecules are simply not encouraged. So the path is set by means of the beautiful specificity that's introduced by the plant enzymes, and this is what really interested us to go after the plant, the medicinal plants and their genomics or transcriptomics. Okay, so with this sort of an introduction on the metabolites, I would also like to tell you we started shortlisting, and from more than 300 medicinal plants that we believe are endemic to India, we shortlisted 25 of them, and definitely Tulasi was on top. Why? Because it is the queen of herbs. And fortunately for us, our collaborator and my long-term friend and colleague, Prof.R.Soudhamini, who some of you know very well, had raw data, sequencing data, both genome and RNA reads. So, which by chance I spoke to them, and the collaboration began. So Tulasi is definitely our plant of choice. And the next slide, I will try to introduce to you about a plant which actually needs no introduction in this country. Tulasi can be related to every household. Many households, people keep a copy of the plant. They even do puja of it because it's considered so holy. And it's holy, and traditionally it's not only holy, but people had realized from the 1st and the 2nd century BC that it is important for well-being of people. To just have a plant like Tulasi in the house means you are ensuring that the atmosphere is kept clean and many diseases can be prevented, right? So this particular text, for instance, it says Tulasi Surasa Gramya Sulabha Brahmamanjari Apeta Rakshasi Gauri Budagni Devadanduvi. These are all the different names that have been given to this plant Tulasi in Sanskrit, and shows how much people have understood that it is an important plant and it's a human's favorite. And I would just like to bring to your attention the last line of this particular shloka, which says Shukla Krishna cha Tulasi Gunyais Tulya Prakirtita. Gunyais Tulya means the properties, the guna is Tulya, it is equal. What is equal between Shukla and Krishna? Shukla is nothing but Rama Tulasi in this case, and Krishna is the Krishna Tulasi. Alright, so this phrase has conveyed the beautiful secrets about not only the value of the plant, of the Tulasi plant, but also how they Krishna and Rama Tulasi can be compared with each other. And still, because of modernization, westernization, whatever, I don't want to give regional attributes here, but you know what all India went through, right? Some years ago, 100 years ago, we went through a lot of changes. And then people of different generation, especially our generation, started asking questions of the obvious. And we would ask things, how do you know that plant Tulasi is really valuable, good for health, and so on. So with this in mind, I told myself, this is an opportunity, given that we are in the post-genomic era and we can do our own sequencing and transcriptome, can we provide a scientific basis so that the future generation can believe these medicinal plants for their value and be able to adopt them early on and prevent themselves from diseases. With this sort of a humble objective, we started looking at the plant tulsi and began by quantifying the extent of metabolites. Okay, the early text said that Krishnais and Tulyas, the both are of equal guna. How does it look when we put these plants in mass spectrometry, in HPLC with all our scientific gadgets? And we find that, for instance, two metabolites, one is called urusolic acid, another is called linalool acid, and I'll come to these in a moment. You can see, as I have highlighted, the Otoliferum Rama tulsi and the Krishna tulsi, they both have nearly equal amounts of urusolic acid and linalool acid. Isn't it beautiful that whatever is inscribed in the Sanskrit text many years ago, thousands of years ago, could be proved right by means of our own scientific measurements? And I hope the future generation will take this as a belief, as a proof, so that they will start to infuse these ideas into their everyday life. Okay, so that's not everything, because quantifying the metabolites is just the beginning. But like I said, we would like to do more. We would like to do genomics, transcriptomics and so on. So this is the botanical description of this plant. It belongs to the order Laminaceae, family Laminaceae, and the genus is Ocimum. The number of chromosomes is believed to be 36. It's a diploid, and the entire DNA content of this plant is believed to be about 612 MB. So with this knowledge, we planned for DNA and RNA experiments. And as I said, our colleague Prof.R.Soudhamini, our collaborator, had these data already. So we started to use these raw data, and during the workshop, what do these terms mean will be brought into better detail. I don't want to go into all the details right now, except to say that these are raw reeds. R1 and R2 refer to reeds, right? And we mixed R1 and R2 reeds, and we got included some more raw data that comes from another genomic sequencing technique called MadePair, which in the workshop we will explain, and please wait for that. It could be tomorrow, yes. And then we used all these raw reeds and a lot of bioinformatics tools that we were learning as we were going along. And as soon as I had got interest on these medicinal plants, I asked my lab members, who were about 20-25 of us then, and I'm fortunate to have at least two of the members here with us who are going to be co-instructors in the workshop. Dr. Abhay Joshi and Dr. Meenakshi Iyer, who are here, right here, and they also volunteered. In fact, the whole lab volunteered, and they all raised their hands and said, we want to work on the genomics and transcriptomics. But the trouble is, none of us knew how these computational tools will work when we started. Okay, so we are bioinformatics labs, but our interests are on protein domain superfamilies, how they are connected, whether we can use genome databases to recognize these relationships early on. But none of us had used any of these bioinformatics tools, but we were all set to do it. So it was a very beautiful process of our learning and trying to deliver at the same time. And we used to set a lot of deadlines to ourselves. We used to have heated debates and very beautiful conversations. And at first we thought we'll meet every month. And then we started meeting every week, and then finally we were meeting every day almost. And we were talking about this all the time during tea break, lunch break, and it's been a very beautiful experience, and almost for the past 10 years. And we are still learning. And in the workshop we will tell you why we are still learning, because there are so many computational tools still coming up in using bioinformatics for genome assembly, analysis and so on. Okay. So this is one of the example flowcharts. In fact, we had to use for sequencing tulsi because the sampling is not enough. Then we have to remove repeat elements, which we do, and then we have to take evidence from transcriptome if we had, and then we also have to bring in evidence, patterns, nucleotide patterns from other plant genomes, and we did all this. And this is one of the flowcharts. For example, we have to adopt. This is a computational pipeline which we use to recognize the genes in tulsi. So getting to know the genome assembly is one thing, starting from raw reads. But the genes are hidden within the genome, right? Not most of the genome is not coding really for genes. And again, we will talk about all this in the workshop. So what does our statistics tell? Although we could kind of assemble nearly 374.8 MB of the nucleotides, which means we have covered about 61% of the genome, we could use gene prediction tools as mentioned in the previous pipeline, by which we deciphered that there could be as many as 36,768 genes. And that's not all, because we also had RNA reads, and that means you can assemble them, do what is called as a transcriptome assembly, and then we did this for Rama Tulsi and for the Krishna Tulsi so that we can compare them, and those values are also mentioned below in this particular table. So that's very interesting, because that is a start that allows us to start doing an analysis. And we asked simple questions about, are we able to find the known genes? Yes, 90% of the known genes we were able to find. And then are we able to find the known domains in proteins that code for particular functions? And these domains are many, like kinases, cytochrome P450 and so on. And you can see, like any other decent plant genome, this particular Tulsi genome that we have managed to assemble also has a large amount of kinases that showed us that we are probably doing okay, because the numbers look similar to the other plant genomes. And we were also fascinated to see a lot of cytochrome P450 in this particular plant, because this Tulsi is a queen of herbs, right? So it must be engaging and using a number of enzymes to produce those beautiful secondary metabolites. And what are those secondary metabolites? I come to it now, for which we did a detailed literature survey side by side, and that means we had looked into nearly 300 publications, book reviews and so on. And then we listed 43 different secondary metabolites, and out of them we could place our hand on at least 23 metabolites for which we knew backend which are all the enzymes that are responsible for producing those beautiful secondary metabolites in this plant of interest. So gathering with this knowledge, we then pin down to, as I said, about 12 to 13 secondary metabolites, and they are shown here. You can see ursolic acid, which we had alluded to right in the beginning when we were comparing Rama and Krishna Tulsi. So that is actually shown as a second entry. It is known to be an anti-cancer compound. Ursolic acid is observed in small amounts in the skin of apple, but it is much more abundant in Tulsi. And therefore, if your forefathers had asked you to chew some Tulsi leaves, it is because of these reasons, right? You know, chewing these leaves perhaps provide you the stamina so that you can prevent yourself from acquiring many diseases. And also you have things like linaloolic acid, the linalool, over there, which is an anti-infective. So you can see how this particular queen of herbs caters to a range of cure against ailments, anywhere from fever to cancer. So you can use it, and that is well known. But coming back to our assembled genome and looking for genes and domains, what we next asked is, for each of these metabolites for which we knew the pathway, this is how the pathway might look like. We start with a precursor molecule, and then an enzyme comes into the picture. It catalyzes and makes it into your product. And then another enzyme comes and it makes it into another product, which then becomes the secondary metabolite of our interest, right? So we, what we do is to search for these enzymes, starting from known enzymes in other plants. It could be in Arabidopsis thaliana, or it could be in a closely related plant, anywhere. So then we use our traditional bioinformatics sequence search techniques to then gather the known enzymes from other plant genomes and go and search in our assembled genome. And when we do that, sure enough and encouragingly for us, we could find a number of hits. Number of hits for each of the enzymes that we know are in the biosynthesis of the secondary metabolites, and in this case we managed to do this for about a dozen of them. And then our bioinformatics really begins, because sometimes when we do sequence searches, and simply because these computational algorithms are very sensitive, you might pick up a number of hits which don't necessarily mean that this is the enzyme that sits in that pathway. So to decipher this, we do what is called as phylogeny. Phylogeny is a way by which you sprinkle in some known members that you suspect your kid belongs to, and then you sprinkle in some distantly related members, so something which is little far away, and then you expect phylogeny to resolve and tell you that if your hit from the Tulsi genome is going sitting in the right cluster, that means it's a true hit. But on the other hand, if it goes and associates itself with the wrong subfamily, so that means it's an interesting gene, an interesting enzyme in plant Tulsi, but this is not what we are looking for, because it's not the enzyme that is there in this pathway, but maybe doing something else. Okay, so for this we also ensure that the functionally important residues are in place, and you can see all these which you see as OTE stands for Ocimum tenuiflorum, which is the Tulsi plant. And these are all the hits that have responded. Isn't it beautiful that you choose a plant that you start to adopt? If you had the possibility to do sequencing, you can get the raw reads, you can assemble them using bioinformatic techniques, you can find the genes using bioinformatics tools, and then you can recognize domains, and then you'll be able to associate with the pathways of our interest. So that is the story which I want to say. And I don't want to just spend too much time on this, because I have 3 other stories. And for me, the 3 other stories are shorter, not as much as the Tulsi, because Tulsi is always very close to our heart. So the second story, which in fact I had talked about, is nothing but Cissus quadrangulis, which is called as perennial in Tamil Nadu. And what does this plant look like? These are different pictures of the plant that we managed to grow in our own lab, and we also managed to take pictures of the plant blooming, fruiting, and so on. So this is a very beautiful experience. And what is of greater interest are the stems. And as the name suggests, you can see the 4-fold symmetry you can see in the stem, and that's why the plant is also given the botanical name Cissus quadrangulis. And I understand in TNAU we have a beautiful rich resource of scissors. We have plants which are having 3-fold symmetry, not only 4-fold symmetry. So I'm sure you have more to say about this plant than I do, than our lab does. But then for us, from the medicinal viewpoint, what is interesting is it is very well known to be used in the case of bone health. Whether people go through bone fractures or joint problems, it has been used for many centuries, for many thousands of years as a medicine to cure fractures. In fact, it is so strongly felt that in Sanskrit, the perennial is called as harjo, meaning something which can bring in a broken bone together, right? So that is the power and proof of this particular plant. And not only that, it is known to also be an analgesic, treat infections, anabolic, promote weight loss, and so on. So what are all the secondary metabolites that confer this property? So that is our question. So the anabolic steroid, the one that is involved in fracture healing, people are still working to understand exactly and pin down the small molecule, but these are known to be estrogenic receptors. And in fact, this particular molecule called resveratrol has been implicated many times for both fracture healing as well as some other ailments. And thankfully for us, this particular plant is rather close to the fruit grape. You may not appreciate it by looking at the pictures, but these are also having tendrils, they are climbers and so on. So they have a lot of properties similar to grape. And the genomes of transcriptome studies results were available for grape, at least for the enzymes that are involved in the synthesis of resveratrol. So we definitely used, so that was our start point. And after we assembled the genome of Cissus quadrangulis perennial and got some statistics that we were satisfied with, we went and searched for these enzymes that are producing resveratrol, not only resveratrol, but we also went after other small molecules such as cetosterol, stigmasterol, and also it shares secondary metabolites as observed in many other plants such as quercetin, campherol. Campherol is also there in Tulsi. Quercetin, as you will see, it is there more in other plants such as moringa. Okay, so that is what takes us next to our third story, which is that of Moringa. Moringa is known to be a miracle tree. So Tulasi is known as the queen of herbs, but Moringa is called as a miracle tree. So these are not given names just simply, but for reasons. And Moringa, for instance, every part of the plant has been implicated to cure one disease or the other, starting from the leaves until the roots. The leaves, as you know, it's used day in and day out to manage diabetes and many other ailments. But the roots, I believe, are used for cardiac ailments. So here people would take some bit of the extracts from roots and use it properly with a proper recipe to cure cardiac ailments. Right, so what we had done in the past is to look closely at this Himalayan tree, Moringa oleifera, and of late, meaning for the past 3 to 4 years, one of my students, Shafi, has been closely comparing this with another species called Moringa concananis. As the name suggests, the Konkan should give you a clue that it is more observed in the Konkan regions, that namely the Western Ghats. And in terms of the geographical distribution, you can see whereas Moringa oleifera is known to be of Himalayan origin, Indian origin, it is spread all around. In fact, some of my friends who are in the US also say that they are growing Moringa. I feel very happy. So it is adapting itself to a variety of climatic conditions. And also Moringa is not only used to cure diseases such as to manage diabetes or cardiac ailments because they have wonderful metabolites, but they also are a rich source of minerals. And many may not know that Moringa leaves offer 30 times more iron than spinach. Can you tell me who in this audience are aware of this? Iron means we think of spinach. How many of you knew that Moringa has 30 times more iron than spinach? Those who know, please raise your hands. Some of my people are and some of my colleagues are, but the students, no, I also didn't know until we measured them. We measured them, and we measured also those of calcium, and it's supposed to have so much calcium, and it offers zinc and so many very nice minerals. And that is actually true for many medicinal plants too. But Moringa is well known to be a superfood. Superfood, and you can give it to people, children who are malnutrition. And that we know in our continent as well as in the African subcontinent, we have many children who are severely malnutrition. So the idea is you can grow Moringa and offer to such children. And that alludes us to the third property of Moringa, which is its strong drought tolerance. It can survive with very little or no water. Right? We cannot survive without water. Most of us is water. But Moringa is not only a miracle tree because it can provide secondary metabolites which can cure ailments. It's not only a superfood because it gives so many minerals, but it also is a very good model plant to show how it can survive in drought conditions. So anyway, so what we knew was this Moringa concananis had higher medicinal value, and this is somehow acquired very little attention. So Shafi, the student in our lab, started to compare these closely using transcriptome studies. So that's a story that I hope to very briefly talk to you about. So the leaves of the Moringa oleifera is shown on the left hand side. The leaves of the Moringa concananis is shown on the right hand side. So the pinnate conditions, they are pretty different. So it's not easy to even recognize that the second one, the Konkana one, which is also called as a wild Moringa, belongs to the same family. And look at its flowers, they are also rather different. The Moringa flowers are pinkish, whereas the Moringa oleifera flowers are pinkish, but the Konkana one are more white, and they have slightly different structures. So this is to show you that the morphology of the two trees are different, but what we really wanted to pursue is how are the distribution of the secondary metabolites different from each other. Can we actually show using in vitro assays that the two plants have slightly different properties? such as, we can use some in vitro assays to show that it is capable of weight management, right? So we can go to that. And like I said before, it's a miracle tree because almost every tissue of the plant is used for one medicinal cure or the other. And we also know, because of our experience in olifera, that different tissues of the tree hold more of certain secondary metabolites. And I had mentioned this in my previous talks, I don't want to repeat it. Quercetin and camphorol are more in the flowers, and then you can see benzylamine is more in the pods, and so on. And this is now surrounded by all these secondary metabolites of our interest. You can see urusolic acid is there, but it is less. And quercetin, on the other hand, is more strongly associated with this particular tree, medicinal tree. And then we did the same, we repeated this for the different tissues of the wild moringa, the Konkana one. And we also could, Shafi could assemble the two transcriptomes using the genome that was published by the Chinese group. And then these values, the statistics values are shown on as a table for the two plants. Okay, armed by these two transcriptome assemblies, we then decided to concentrate on only 3 metabolites. Like how starting from 43 metabolites, we concentrated on 12 of them for tulsi. To compare the two types of moringa, we decided to concentrate on only 2 secondary metabolites and the enzymes that are involved in the synthesis. Quercetin. And there are as many as 6 different, in 1, 2, 3, 4, 5, 6 different enzymes that are involved in the catalysis of the initial product, the primary product, going from one to the other, acquiring its chiral centers and so on. Finally, it gives rise to the quercetin, right? And then on the other hand, benzylamine. The molecule looks so simple, but it is very powerful and it is found in abundant conditions in this particular plant. It has another name called morinjane. So you can imagine how much this benzylamine is associated with this particular tree. And we also decided to focus on a third secondary metabolite called chlorogenic acid, for which as many as 9 different enzymes are involved. And in particular, we focused on HQT, HCT, because these are very similar enzymes. They are more than 30% identical. And at some stage, if the HCT is more, it can give rise to a certain secondary metabolite. If the HQT is more, it can give rise to another metabolite. So you can imagine how much the presence of these enzymes will contribute to what sort of secondary metabolites will come. So for what does this heat map say? The heat map is no longer concentrating on the medicinal value, the secondary metabolites and so on, but instead it is showing us the relative abundance of the enzymes that are back in the synthesis of the 3 secondary metabolites that I said, quercetin, benzylamine and chlorogenic acid. And since there are 8, 2 and 9 different enzymes involved, this heat map is a way for us to compare and appreciate how not only the two plants differ from each other in harboring the enzymes of interest, but also how the different tissues, namely flower, leaf, stem, root and so on, are holding the different enzymes. These have been carefully calculated and these are called as TPM value, total parts per million of the RNA reads that are observed in the different tissues between the two plants. So whichever is dark green means it's more abundant. The lighter the green color, the lesser the amount is. As you can see, the last 3 enzymes, the second, third and fourth enzymes in the quercetin pathway, it's much more abundant in the stem of the wild moringa. So this, we think, is kind of a minor breakthrough. It is an encouragement for us to believe that this may be some of the reasons why the wild moringa has better medicinal properties, because it has more of the enzymes that are required for that secondary metabolite, quercetin. And quercetin is well known for its anti-diabetic properties. And what I am not able to go through the details, but for all of these enzymes we have carried out what are called as QTR, PCR, by which RT-PCR experiments by which we can quantify and validate that indeed these enzymes are observed in large amounts in the tissues of the interest. And you can see that these values are corroborating very well with the RNA and the transcriptome assembly that has been used to compare the two plants across the tissues and so on. Okay, I think I will just conclude the story by telling you that we have indeed carried out not only the quantification of the metabolites, and you can see what is shown in dark purple color is the wild moringa. And you can see that chlorogenic acid, for instance, the amount is as much as 244 in the wild moringa, and it's much less in the moringa oleifera, where you see only 171. Shafi has also carried out in vitro assays that directly impinges on a better understanding of the lipid management and how it reduces obesity in humans and so on. But almost all this comparative analysis I have talked about are unpublished. We are preparing the manuscript, so hopefully in my next visit I can share some of these details with you when hopefully we will have published. Okay, so that then takes us to the last story, and this is also the one which is the most recent. It is the story of Sankapushpi. Sankapushpi is again not only an ornamental plant, it is a plant that is used in religious situations, and it is a very holy plant, and the botanical name is Clitoria ternitia, and it is been used again for thousands of years for several medicinal reasons. It has been known to alleviate microbial infections, act as an insecticide, and it is cytotoxic, anti-inflammatory, anti-cancer. And this picture kind of emphasizes the original and the most prominent reason why the Sankapushpi flower tea is used by humans, especially in some tribal populations. People still use them when a lady is going through childbirth. The childbirth is associated with huge amount of uterine contractions and pain, so it is believed and documented that consumption of this tea reduces the pain and also eases out the childbirth. And back end, what we know from the scientific angle is that the plants produce this sort of special secondary metabolites, which are also called as cyclotides. There's something very, very special about them. They are not small molecules like the quercetins and the benzylamines and the chlorogenic acids, uracilic acid which we saw before. These are themselves proteins. And these are also held together by covalent cross-links called disulfide bonds, and every time you see a C, that means in the primary structure is a cysteine, and that means this C and that C are committed into a covalent cross-link, a disulfide bond. And you can see that there are lots of these cross-links going on, like a metro map. So that means there are many disulfide bonds. And every time there is a disulfide bond, the covalent cross-link, the protein is said to become stronger and stronger. Stronger against what? Against thermal stability. Because proteins have a tendency to denature, right? If you boil the milk and then you overdo everything, then it can get denatured. But these cyclotides, on the other hand, they are known to be stable even at 120 degrees centigrade. And this is possible because it has 3 disulfide cross links, and also it cyclizes. Cyclization also gives higher thermal stability. So the N and the C-terminus are forming a peptide bond. So this is the beauty of this cyclotide, and there are several structures known, not of the cyclotides in Sankapushpi so much, but from other plants. And this is the cartoon diagram of one of the structures of cyclotide from a related plant. And what are all the plants that secrete cyclotides simply for, they do it for their chemical defense, right? And that's how we started the discussion, isn't it? The secondary metabolites are produced by plants because they cannot run away from pests or herbivores. Okay, so all of these plants that are shown here are examples where they naturally produce cyclotides, and one of them is, you will be interested to know, is nothing but coffee. Coffee, and in fact one of the species, I am sure some of you can relate, that you have a coffee variety called Robusta. Robusta because it's robust against pests and herbivores, and because the Robusta coffee produces more amount of this cyclotides. Right, anyway, the structures are known from one of these. I will not go into too much detail except to tell you that a good amount of structural information are available from all these plants, and for each of them the secondary metabolite of interest are the cyclotides. And like some of the conus toxins, the cyclotides not only are proteins but they have genes behind them. These are not like the small molecules which depend on enzymes to get them synthesized by the plant, but there are genes encoded. There is a pre-pro-cyclotide which gets cleaved and cleaved again until it is ready to produce its and kind of present itself as a primary defense for the plant. Okay, so that is the other fascination. So where are we doing with respect to our transcriptome analysis? That's what I want to tell you. One of my collaborator student called Neha decided to focus on this Sankapushpi and extracted the cyclotides. Why the extraction? Because first of all, these are proteins that behave as secondary metabolites, and also these are thermally highly stable. So she would just boil the plant, the leaves and flowers and all, and then go after the traditional extraction procedure, which gives a large amount of cyclotide, which she then used it for transcriptome sequencing. And in fact, she took other tissues as well, flower, leaf, or stem, and then she would take the RNA and try to purify it and take it forward, and she took it forward for transcriptome assembly, much like what was done for moringa. Right? And here I had very expert collaborators, Dr. Radhika Venkatesan, who was in NCBS like a year ago, and she had expertise not only in handling plants and extracting metabolites from them, but she was also particularly interested in plant-insect interactions. And then I also was fortunate to have another collaborator who is none other than my PhD thesis supervisor, Professor Balram. And as you know, you may know, Professor Balram is a chemist, and he is very interested in small molecules and especially structure determination using mass spectrometry techniques, and that's where he contributed. And together we started asking questions such as how many cyclotides are there in this plant of interest. And we found that we pick up all the known ones, and also we pick up some unknown ones. Some normal cyclotides which we see only in this plant, and that's understandable because if the plant is very effective herbivore, it must be producing a large number of cyclotides, different types of cyclotides. And that is shown here as some of them which are actually novel and present only in this particular plant and so on. This also shows you the pre-pro arrangement of the genes of interest. So that is about the cyclotides, but we didn't stop there. Whereas we don't have enzymes which produce the cyclotides because they are genes themselves, we asked, because these are disulfide rich systems, what about the allied enzymes which are known to help, such as prolyl oligopeptidases, thioredoxins, and also the fact that it is a cyclic molecule, that means N and the C-terminal amino acids form a peptide bond. So there is a particular enzyme called aminopeptidase which is used as a catalyst. So we went after these questions, and we found, for instance, we can evidence novel cyclotides. Using mass spectrometry, we were able to attribute its sequence and enlist them, and there were more than close to 100 cyclotides in this beautiful plant. And apart from the cyclotides, like I said, we concentrated on enzymes that facilitate disulfide bond formation, such as protein disulfide isomerases, which are used as catalysts in different parts of the cell when it is the disulfides are vitally required to form and give the thermal stability to the protein of interest. And we found that several PDI, as it is called in short, in fact are endemic to this plant. And we see that a number are co-clustering with the known PDIs from other plants, But one or two cases are shown in these ellipse here are observed only in Sankapushpi, suggesting that there must be a very specific requirement and recruitment process by which different PDIs are simultaneously expressed and recruited in different parts and tissues of the plant of interest. And likewise, we also ask the question whether we are able to find the enzyme that is used for the cyclization event, because the cyclotide is cyclic, right? And this is commercially very important in the industry, because many people would like to cyclize peptide of many types for different reasons, and they always find it very hard to know which is the enzyme that will do it. So when we presented it in different places, people were more interested in this enzyme than the cyclotides themselves. So anyway, we were grateful to find a few entries of this particular enzyme, and in fact we found multiple copies of the many transcripts of this enzyme in this plant, and these are also shown as a circle. And like I said in the beginning, and we will allude to it in the workshop later on, only if it co-clusters with the accepted other enzymes coming from other plants do we accept them as a true hit. And in fact these hits co-cluster very well with other known asparaginal endopeptidases. And therefore we are now convinced that these are perhaps the true enzymes required for that reason. Okay, so with this I think I will really stop this fourth story on the Sankapushpi. Both our stories on Tulsi and Sankapushpi acquired a lot of attention because it reaches the community, right? So we went on press, and especially for Tulsi we had a very pleasant experience, because I found myself getting interviewed in different languages, first in Hindi, next in Tamil of course, and then Kannada, and then finally in English. So our team had a lot of fun doing all this, but what I want to do now is to tell you who are the people involved, my acknowledgements. Like I said, we started off with Tulsi, and when I said shall we work on plant genomics, the entire lab raised their hand for volunteering for this new work. So we are all there. Plus, none of this would have been possible without the very nice lending hand from Prof.R.Soudhamini, and also the CCAM colleagues, Dr. Malali Gowda, who was himself a plant genomics expert. And this is these are the people involved in Tulsi, in perennial. We had our lab. Okay, this is now capturing ourselves in action. Like I said, we used to get together and sit and discuss almost on a daily basis. You can see several people sitting here and discussing. Here is a bike. Somewhere Meenakshi is also there, I'm sure. Yeah, so you can see that we are all sitting and discussing and trying to understand all this. Okay. Whereas in the Moringa, we had reduced to a small set of active people, but all of them had actually handled the raw data. And you can see definitely Meenakshi is here and Advait is there. And Shafi, who is a person who is looking at this cross comparisons of two types of Moringa, the wild one, is here. He is the one who is very quiet but very efficient, because he not only did the transcriptome, he would run around, go to Transdisciplinary University, which is an Ayurvedic, which has an Ayurvedic institute, and carry out the wet lab assays as well. And he will come online for the workshop. And we also had for the Sankapushpi a different team, like I said, Neha, who is a very, very effective student, who did all of these works, the transcriptome work, the mass spectrometry work, and the behavioral studies in worms, which I have not talked about. And plus she also updated our disulfide database. She was very efficient. She managed to shuttle between 3 labs and kept smiling. And these are some of us who kept troubling her, I suppose. But no, I was very fortunate to have collaborated with Radhika and Professor Balram on this project. And last but not the least, I would like to stop by saying giving my acknowledgements to none other than my mother. My mother, because she was a person who used to give away tulsi seeds as a gift to people, and how much of foresight she had. And she had actually induced me to think about plant as medicinal units. And also she was the one who told me that there is some other species of moringa called wild moringa which you have to go after because they have conferred better medicinal potential. And thank you to my mom for including me. If you have any questions, I'm happy to take at this point. So these next two slides will provide you some glimpse about what the workshop is coming about, and Dr. Abhay Joshi has prepared these two slides along with some introduction on what is we would like to know from the bioinformatics sides on the genomics and transcriptomics, and I hope it will also help you to get prepared and prime into learning a lot more about the bioinformatics aspects of doing computational genomics.
To look for the bioavailability of those nutrients or the metabolites, because we are applying on the wounded person. So instead of swallowing it, we apply it on it. So will it be okay to look at the bioavailability perspective?
Yeah, I think if plants provide them, human body is not able to absorb all of it. And that is true, that may be well known for turmeric and the curcumin that plant offers. But even here, the bioavailability is an important question. And in fact, we could add it along with other, time and again it has been proven that if you mix it with certain other compounds, the bioavailability increases. In this particular case, it may be a different one, but in general, people notice that pepper and honey provide a better bioavailability for the secondary metabolites. So instead of focusing on the enzymes and the commercial aspects about being able to synthesize them in large amounts and so on, what one can also do is to provide better application in terms of providing better protocols of using these plants. And one of them is bioavailability. And then another is convenience of applications. That could be, because some of these ideas could be patented. And for instance, if you have strips by which you can just put as a band, along with a band-aid, can this stay on and provide the better healing properties. We could also have some surface entropy increase so that important molecules are held and provided to the tissues in better efficiency. So these are some of the open questions for which we need innovative ideas, and maybe some of the young minds will come up with them.